Thank you very much. My name is Dr. Denis Bauer, and thank you for the invitation to the chairman and the organizing committee. What a wonderful event to commemorate the 40 years anniversary of SCL. So allow me to start by introducing the research organization that I am part of, which is CSIRO, which is Australia's government research agency. At CSIRO, we're quite unique in covering the full value chain from basic science all the way up to bringing technology into the clinical practice. The tools and technologies that we have entered into the clinical practice, the most famous one is, of course, Onto Server, which underpins the digital health agencies around the globe, as well as CardiHub, which was the first mobile health app for heart rehabilitation, and Hotspots, which is a digital surveillance that helps doctors choose the right drug for the right bug at the right time. But today I want to talk to you about genomics. And hopefully by the end of the talk, I want to have convinced you that every single one has a mutation that should influence the clinical care we receive. But the problem with that is that knowing which mutation for an individual is the actionable one is really difficult. And that is the main thesis of our recent Nature Biotech paper, which argues that we need to bring data together from around the world to really understand what is the genetic diversity of human beings in order to identify what are the true disease-causing genes. But before we go there, I want to tell you 3 stories. So the first one is around understanding disease risk from infectious diseases and non-infectious diseases. The next one is actioning genomic insights, and specifically with robust interoperable systems. And the last one is around going beyond to the next generation of global collaborations. Good. But before we go there, allow me to bring us all on the same page when it comes to finding disease genes. So here I have lines that represent individual genomes. The bars, or the little boxes, represent individual mutations that each one of us have. So on average, there are 2 million differences between you and the person sitting next to you. And for disease research, we identify a cohort that has a certain disease and compare that to the normal control, so healthy individuals, with the aim of identifying individual mutations that are enriched in the disease cohort and ideally absent in the healthy controls. But the problem is that the genome is huge, and usually the effect on complex diseases on the outcome of the disease is very small. So the bar here represents what we typically identify as genome-wide significant, and hardly any mutations really reach that genome-wide significance. So therefore, people go about and saying, well, how about multiple positions in the genome working together? Surely that will explain more of the genetic risk. And that is true. But the problem really is with complex diseases, it doesn't work in an additive way. It is a much more complex approach of areas in the genome that exacerbate the disease, but areas of the genome that also are protective. So we do need to use a machine learning approach in order to identify these genome-wide associations in the genome to truly understand what the disease risk is for an individual. And that is what we tackled. So our approach is we take a case control cohort. We use VariantSpark, which is our machine learning method, in order to identify the genome-wide associated joint interactions in the genome. And then we use a second tool called BitEpi to then identify which of those interactions actually work together. So which genes cause the disease together in a network graph that I'm showing here on the left side to truly understand the etiology of molecular disease. So here I'm showing the paper in case you're interested in looking at more of the details. But suffice to say here is that the key to VariantSpark being successful is that it can run much faster than all the other machine learning methods in a similar domain. And this allows us to apply it to large cohorts of thousands and tens of thousands of individuals to dive in and identify the small but complex signal into the genome. A key element here is using machine learning in the disease research was around distinguishing noise from the true signal. So we developed a new statistical approach specifically for genomics and machine learning in order to identify that threshold that separates the noise from the true signal. And then, as I was saying, BitEpi, the key to BitEpi is, again, that it can run faster. It does an exhaustive search on all the pairwise, triplet, and four-way interactions into the genome, and it is the first method to do so, which, of course, enables us to truly identify what are the pathways and the interactions in complex diseases. So we applied VariantSpark BitEpi combination to the UK Biobank, 50,000 case controls in cardiovascular disease. And we identified signal that typically would take 10 times more signal in order to identify. And this really opens up the door to beautifully annotated but smaller data sets to help elucidate what genes actually contribute to the disease. And an example of that is Alzheimer's disease, where we just applied it to 780 individuals. And here we were able to identify 2 novel replicating genomes, or genes, that could really explain in a much more detailed way the molecular differences of Alzheimer's disease. And of course, in motor neuron disease, we applied it to 10,000 case control individuals and identified novel genes in the Australian population that were specific to the Australian population. And this was together with the international cohort, Project 9. But the key here, I think I want to highlight, is that in order to truly understand the genetic differences, we currently have only European data available, which of course isn't good enough if we want to treat the world's population. So here the GWAS diversity meter is demonstrating that currently it's 90% European genomes and only 10% others. And hopefully over the next years, this shift, or there is going to be a shift, so we can truly understand the genetic diversity of, for example, the Korean population. And just to drive that point home, is we looked at cystic fibrosis and carriers of cystic fibrosis in the Australian population. And what we identified is that currently the gold standard of identifying cystic fibrosis carriers is truly adapted to Europeans. So therefore, the East Asians that we looked at, we were missing with the gold standard 44% of the cystic fibrosis carrier, which in my book isn't good enough in a population that is very diverse. So I'm quite delighted that I was invited with SCL to work on a Korean cohort, which exactly looks at those hypotheses, that there is a Korean-specific marker of cardiovascular disease. And therefore, if we want to treat the population properly, we do need to identify the data and analyze the data of the right population rather than having the Europeans as a proxy. And that brings me to the second story around actioning genomic insights. So you might be familiar with the Beacon protocol, which is a protocol of sharing genomic data that is accepted around the world. And the concept here is that you look at, or you send out queries to cohorts to ask, have you seen a particular mutation at a particular position? And it comes back with a yes/no answer in order to preserve privacy. And the idea is then that you can link up multiple cohorts around the world in order to ask this question, not only of one cohort, but all the diverse cohorts around the world. And this is exactly what we've done with our Nature Biotech paper. And we've taken this beacon protocol, which wasn't very scalable, and we brought it into the 21st century in making it cloud native, with the idea that not only can you query global cohorts around the world, but you can also query the medical ontology data. So here we partnered with OntoServer, which I said earlier is underpinning the digital health systems around the world by bringing in SNOMED and HL7. So we, for the first time, generated or created the ability to query genomic data alongside medical information. So for example, you can ask for individuals with mutations at a specific location and also specific features, for example, neuroblastoma. So this enables the global research community to do phenome-genome queries in real time. So in just 88 seconds, you can query vast genomic cohorts of millions of individuals. And you can do that at a sustainable cost. So for less than a cup of coffee per day, we enabled consortia around the world to contribute their valuable information of those individual rare population cohorts to bring that information to the world. So again, we hope to apply this here at SCL, where we want to query then, or we want to share the information that is identified in the cardiovascular disease specific to Korean population, so that the rest of the world, the rest of the Korean populations around the world can benefit from this information. Now, changing track here a little bit, because so far I've been talking about human genomics. But all of this, of course, applies to pathology, pathogen genomics as well. So taking, for example, COVID, which we know has originated from an animal reservoir somehow and jumped to humans. And as it does so and jumped from humans to humans, it accumulated more mutations. Some of those mutations made it more infectious. Some of them mutations potentially has the opportunity or has the probability of making more virulent. So therefore, being able to analyze those individual strains and predict their features is absolutely critical. So we developed a machine learning method that can basically put can generate a genetic fingerprint of each individual strain and then put it on a 2D map in order to visualize the differences between two strains. So we applied this to COVID way back when there were only 181 strains available. And the aim there was to identify the first vaccine, or the strain to which the first vaccine needed to be generated for. So the task was to find something that is current, but that is also going to be relevant in the next years or months and years to come. And therefore, being able to extrapolate from there with this approach was absolutely vital. And going one step further, we then asked the question, well, are there in the genome mutations that can predict the outcome of the disease? Because we know that some people don't have any symptoms, while others suffer severe symptoms from it. And therefore, we applied the variant-spark bit-epi combination to 5,000 cohorts around the world, some of which had severe disease and some had mild symptoms. And we identified 100 locations in the viral genome that are predictive of the outcome. Some of them were in the spike protein, as to be expected, but some were outside in a gene, NSP14, which has now demonstrated to modulate the human immune system, and therefore, of course, it has an impact on the disease outcome. So we partnered with Indonesia in order to then be able to extrapolate from this information. The new mutations, the new strains that are coming, can we put them on a visual map? And can we preemptively identify which ones are going to be the difficult ones, the ones causing disease, and which ones are just the endemic one we don't need to care and we don't need to worry about? So one element to that was really important was the data sovereignty. Because individual labs in Indonesia, and I'm sure around the world, they have an investment in their data that they generate. And therefore, being able to share that data with the rest of the world is not necessarily something that their data access policy allows. And therefore, we developed a methodology that allowed the data to be retained and only the insights be shared with the world. So you still can treat it like one global outbreak, but you don't have to give away your data. And this brings me to the last story around going beyond, because everyone wants to work together. Everyone wants to have the data from other people, but there are difficulties around sharing the data, technical difficulties, policy difficulties, and therefore we do need to go beyond that. And one element will be to develop the right technology. And just to drive home how important it really is to have access to the right data, what I'm showing here is the annotation of COVID as it was made in GIS8 around the disease outcome. And as you can see, in May 2020, the vast majority was not known. So the individual strains that were submitted to GIS8 were not known what kind of outcome there was in the human population. We then worked together with GIS8 in order to make that field mandatory, and therefore, rather than not provided, it now was annotated as unknown, which wasn't much helpful either. But working with the pathology providers and changing the workflows at what point they provide that information to GIS8, we were able to improve the annotation quite a bit, as you can see. The problem, though, with that is that GIS8 implemented a free text form. So all the pathology providers around the world could enter whatever they wanted, and that made it very difficult to analyze. So, for example, the loss of sense of smell could be annotated in vastly different medical terms. So again, it's SNOMED CT and its hierarchy that is helping here, but if it's a free text form using ontologies, you can't do. And therefore, we worked together with GIS8 and came up with a questionnaire, an automated HL7 FHIR-based questionnaire that allow people to type in free text, but under the hood, it would be converted into SNOMED CT terminology. And again, this is together with the Onto server. So therefore, the trends really that I see in the digital health space is interoperability is going to be key. We do need to have the data in a way that machines can read it to analyze it at a big scale. The next point, of course, is personalization. That is the opening statement that I made of saying everybody has mutations that should influence their clinical care. And I would argue that bringing data into the cloud enables this personalization, because now you're not only having access to your patient's information, but can compare it to the patient information from other cohorts. And then, of course, developing data analytics as a service in order to truly collaborate internationally, where we developed methodologies in Australia that can then be rolled out as a service to the rest of the world. And that is basically what we've done with the public cloud providers, AWS, Azure, and Google. And so yesterday we heard that SCL has lots of firsts, and I can relate to that, in that being the first in a certain area is a very rewarding position to be in, because you can shape the future. So in 2016, we were the first bioinformatics tools to demonstrate that you can be cloud native, that complex workflows like research can be handled by cloud technology. In 2019, we went one step further in being the first public sector organization to have put a product on the digital marketplace. In 2020, I became a data hero, which is a distinction of cloud practitioners around the world, and there are only about 2,000. And in 2022, we replicated the achievement that we had with AWS in bringing the first product onto their marketplace, with bringing the first product onto the Azure marketplace as well. So Variant Spark, our machine learning tool, is now available on 2 marketplaces depending on where data is located. And just to drive that point home a little bit more is that, to me, putting something on the marketplace, yes, you can commercialize through that, but I think the key element to that is reproducible research. Because we can develop the environment, the analytics, and the data handling that is reproduced all around the world in exactly the same way. And therefore, the outcome will be exactly the same way. And therefore, when you point it to your data source, you know that the only difference is your data, and the rest is exactly replicatable. And that is a key cornerstone, I think, of medical research. And just to finish off on the flip side, if you want, of being able to accumulate data at this massive scale means that we need to accumulate it in an ethical way. And to me, this means dynamic consent, that individuals have the ability to consent to the research done, but also in a dynamic way to retract their consent going forward if something changes in their situation or if they become aware of certain practices. And therefore, we developed a fairly complex framework of how dynamic consent should be handled in the future, using distributed systems, using the cloud, using ontologies in order to make this a reproducible, fair, and equitable approach that puts the patient at the center of the process. With that, thank you very much for listening, and over to you.